Buddy, this is the walkthrough video for our Tuesday and Wednesday bell work. Remember, unless you're taking the SAT, the expectation is you'll do some schoolwork on Wednesday, no matter where you are. And so we're starting this assignment today. You know the drill. Whatever you don't finish does become homework if you don't finish it in class on Tuesday and Wednesday. So this is the last thing we have to learn for our unit. It's really important. How do you know if a trait is determined by genetics, the environment, or both? That's what I should probably say here. Genetics, environment, or both. So we're going to analyze real life data about a plant we use in the Americas for sugar. And the assignment's called Sweet, Sweet Science. It's an online assignment because you're going to be learning how to make some graphs using a spreadsheet. Some of you have had a lot of experience doing this in middle school. For others of you, it's your first time. No big deal. I'm going to walk you through it in this video. So, where have we been? You've seen this slide. We can follow traits from ancestors to descendants, and we can make predictions about future kids using Punnett squares and pedigrees for dominant and recessive, for multiple alleles like codominance and incomplete dominance, and for traits that show up on the X chromosome, those genes for the alleles for those genes are on the X chromosome. And we know that you only get one chance at anything on the X chromosome if you're an XY person, and then that changes XX and XY phenotypes differently. So the one part we haven't talked about yet is like, where do new alleles come from? Most guinea pigs are hairy. And so like, where did hairless alleles come from to begin with? We learned this week that new alleles come from mutations and that different types of mutations have different effects on the proteins that are made from the DNA code. We learned about deletions, insertions, and substitutions. It's red because the computer doesn't recognize A, T, C's, and G's as spelling. That's great. We learned about frame shift mutations, missense, silence, and nonsense mutations. So those different ways of thinking about what happens as a result can really change proteins. And you saw some examples of those in yesterday's assignment. So you actually have this case study about the blue people of Kentucky. I think I talked about it a little bit yesterday. Once you finish today's assignment, you're going to be in much better shape to finish this practice study case problem. But like blue colored skin, which it turns out was a really interesting trait from these people. This is a false color photograph, but does give you the idea of just how blue people's skin were. It was like obvious to people. Oh, sorry, I'm yawning. I'm recording this really late. Another example, there is a remote village in the part of this world where there was a family, and there was a biological family of two parents, and they had many kids who walked full time on their hands. And that has been explored. But it's like a really interesting question. When we see big phenotypes like this, like how can we analyze data to tell if a trait difference is caused by genetics, just the environment, or if it's like a combination of both? Like what makes phenotype? Is it genes? Is it environment? And is a combination of both? And I'll be honest with you, for most of the phenotypes that we think are interesting, it's definitely both. Even the people who would be the best at track and field or swimming, if you don't get to practice throwing a javelin or being in a pool, then you will never know if you would have been a phenomenal swimmer, right? You need that environment and you need practice to get better at it. Here's another example in plants, which is our focus today. You might not know this, but if you do 4-H or if you have family who does farming, we think a lot about how we're going to feed future people, and thinking about plant growth. So imagine these are two gardens. They are farm plots that are right next to each other. They have the same amount of rainfall, the same amount of lighting, and I've planted the same plant in them. These are the exact same bunch of seeds from the exact same parents. But look, this soil has lower nutrients, and this soil has like a typical nutrient. So remember, these plants, carbon comes from the air from photosynthesis, but nitrogen, phosphorus, and sulfur, plants need those too to build their macromolecules. And so it's quite obvious that there are still differences in this garden or this plot area where there's no low nutrient. There are differences between the heights of the plants. Same thing here. There are differences between the heights of the plant. But if you compare those two groups, there's a difference overall as well. And that's kind of what we're getting at today. We're thinking about analyzing trait using averages to tell if a trait difference is caused by just genetics, just the environment, or both. And the plant we're going to use is sugarcane. It's a plant that we harvest to make sugar. We also harvest sugar beets, but this is sugarcane. We press it, we extract the juice, and then we crystallize it to make what you think of as white sugar. Sometimes there's a bleaching process involved. And so hopefully it's obvious to you that farmers want to grow as much sugar as possible, which means they want plants to grow bigger. And it's not just about food. We also use sugarcane to process to make fuels, biofuels. And we're looking at biofuel replacements for when solar, wind, and batteries are just a combination that might be a little less practical. So lots of different interesting things. Today's assignment is online only. You cannot do a paper copy of it. So go ahead and watch the video with me as I tell you a little bit about how to do this assignment. So you're going to see the assignment here. You need to be signed in, so it needs to be your picture up here. And then you're going to go to File and Make a Copy. And when you do that, it's going to show up like this. You're going to make your own name for it. Like, do this. You're going to make sure that you type in your first and last name. You're going to go up here to sharing, and you're going to set it here. You have a choice that's going to say anyone in Monroe County Community Schools. That's the right one. Then the last thing you're going to do is copy the link and submit this assignment to Canvas. So you're going to hit Control Copy. You're going to go to the link on Canvas that's right above this video or below this video, and you're going to enter that link as the assignment. Now you can do that right away. I'm not going to grade these until I come back. And so you can keep working on it over the next couple days. All right, let's get into the actual assignment where we think about Venezuelan sugarcane. Researchers from Venezuela, we have a lot of people in our community who are from Venezuela, people who are recent immigrants, as well as people whose ancestors came from there. So it's an interesting place to think about. Researchers from this country conducted investigation on sugarcane production in their country. They planted a number of varieties of sugarcane, like those shown here, in different regions in the country. And I bet you can already tell what some of these outcomes are going to be. So this row of plants has a different genetics from this row of plants, who has a different genetics from this row of plants. They were interested in how genes and the environment would affect sugarcane production. So here's a country of Venezuela. Oh, big yawn, sorry. And here are the 5 places. Hopefully you can see they're going to plant different types of sugarcane that have different genes, but these are also very different environments. They have different soil texture. Loam is the type of soil that is really great for planting plants. You can see, though, that there are different types of grit size in these loams. They get different amounts of precipitation. Imagine that this is 7 and this is 15. This place gets twice as much rain per year as this place. And then the soil pHs are different. So a pH of 7 is neutral. So these two places have a pH of soil that's about neutral. This place has a really acidic soil. These places have pretty basic soils. And so all of those will combine to make different environments for each of these locations. So what you're going to do is take a look at this data and think about, if it's just environment, where do you think sugarcane is going to grow the most in this investigation of these places? Where do you think it's going to grow the least? Now, they're all about the same latitude, so they're all going to get about the same amount of sun. So you're going to explain your thinking. You're going to name a region here and explain why, name a region here and explain why. So pause the video and do that. Make sure you use one full sentence. Okay, now let's take a look at the research. These researchers planted and harvested 6 varieties of sugarcane over 2 successive seasons, just to control for like extreme rain or extreme drought. The sugarcane was placed in randomly assigned blocks, and you can see the average yield here. And this is 2 cycles of harvest. So you really are getting a big data set. If you click here, it's going to ask you if you want to make a copy of the template, and you do, and it's here. And I've already gone ahead and made 2 tabs. So you'll see there's a tab that's called Sugarcane Harvest by Location and a tab that's called Sugarcane Harvest Data by Genotype. Let me show you what I'm going to do. It's going to go a little quickly, so just make sure you rewind the video if you have to. So what we're going to do is have the computer take an average, and you actually don't need to do a lot for this. The computer will help you think about it. You'll see that I've bolded the different names of places. We're going to get an average for each of those places. And then on this tab, you can see I've bolded the different genotypes, and we're going to do the x-axis as genotypes here and think about whether or not we can see a difference across all the places. So in order to do that, I'm on that original tab. I'm going to hit equal. I'm going to type out average in all caps. I'm going to open parenthesis. This one's for Santa Lucia. So I'm going to take all of those like that. I clicked on the first one and held click while I dragged them all down. I also could have typed in the range with a colon, so B2 to B7. I'm going to end the parenthesis and hit return. And it actually is even giving me an autofill suggestion that says it's going to go through and take the averages of all of those places. What formula is it making? It's going to make the average of all of those individual spots. So just to make sure that it worked correctly, I'm going to type here and I can see it didn't work correctly. So in order to make it so it happens, we learned do not autofill. Instead, I'm going to type average. I'm going to get this row of numbers for that place. And then I'm going to type in this next row of numbers equals average. And it's going to be this row of numbers, right? So go ahead and do that because you won't be able to just watch my screen. You're going to need to pick those up. So go ahead and pause the video. Great. I'm going to go ahead and do the rest of these just in case it was not clear how to do it. I'm dropping and dragging for this place. Then I've got this. I'm going to drop and drag for this place. Great. So now I've got this x-axis situation where along the x-axis, I'm going to make a bar graph. I'm going to show how much yield there was in different places. So let's do that now. We're going to collect all of this data. So I'm going to highlight all of that. I'm going to go to insert and chart, and I'm looking for it to make a bar graph, and it did. It helped me remember as a double check to make sure my x-axis and y-axis are correct. But now you need to go in and you need to type new labels. So all of these are the different places. So I'm going to type locations here. And then the y-axis is the amount of sugarcane, right? So let's go back to the assignment and see, here we go, and see it's tons of cane per hectare. So I'm going to copy where it says average yield, tons of cane per hectare. I'm going to use Control C to copy it. And then when I go into this place that says x-axis, I can leave that label and hit return, and that's the appropriate axis label. Now I'm also going to need a good name here. Happily, it'll tell you what that y-axis versus x-axis is. So I'm going to type in locations. Oh, sorry. I'm going to type in average yield for each. And then the x-axis is location. So that's a really good name of this graph. And you can see already maybe you can decide where did you see the most? Did it match with your prediction that you made on the assignment? Where is there least? Now we can't just do that. We also have to take a look at the different genotypes. So if we see big differences here, that might tell you that genes have something to do with it. So this time I have to take an average across because I'm averaging across all the places. So I'm going to do that same thing again. I'm going to type average. And then I'm going to open up that parenthesis, take the row and hit enter. Now I think this one's going to autofill correctly. So let's find out. When I type here, it shows that row. When I type here, it shows that row. That's great. Go ahead and pause the video and do that. And then the next thing we've got to do is go ahead and make a a chart. There we go. And it is going to go ahead and make a bar graph. This bar graph is blue like the last one, but it is showing something different. Remember, we're looking at the x-axis and the y-axis. This time the y-axis is still the same, so you can go in over to your other graph. You can just double click on all of that and hit Control C, and then come in here and you can type where it says y-axis and you can hit return and that'll be the correct y-axis. But your x-axis is different. This one is genotype, right? And so when we do the name of it, we're going to say average yield for each genotype. right? That's a different graph. It's showing you something different. And hopefully you can already see that there really is one genotype where you see a big difference. I don't know if you know about error bars, but if you've ever seen those on a graph, that's what we do next, but we don't do that in on-level bio. So the next thing you're going to do for your assignment is it says organize the data in a graph that allows you to investigate expressed traits in the 6 varieties of sugarcane. The 6 varieties of sugarcane is the one by genotype. So you're going to copy, Control C, and Control V. Link to spreadsheet, doesn't matter. I'm going to choose to not do that. So that's the graph where the average yield for each genotype. And then for this one, we're going to take a look at the one that is sugarcane by location. So I'm going to hit Control C. It says here, calculate the mean for each area, for each place. That's what we did here. Pardon me, I have stuff all over the place down here. Okay, great. So I'm going to zoom out a little bit, and I'm actually going to view this document. I need a print layout. I need a print layout. Is it mode? Show print layout. I kind of want to show this two up. Let's see if I can do that. I feel like usually this is easy, but I'm very tired. Can't do it. I'm too tired to do it. Sorry, friends. If you can do that, I'm so happy for you that you can do that. But we're going to take a look at these two graphs side by side. And I think the way I'm going to do that is just to zoom out so you can see both of them together. So there's one of them. This is per yield, and this is per genotype. Is there a genotype that makes a big difference? Yeah, whatever happened here, you get a lot less sugarcane than you do with the other ones, quite a bit. Like the difference between here and here, that might be the difference between here and here, but then it's even bigger drop, right? And then let's take a look here. Which locations have the best growth for sugarcane? So that answer is what you're going to answer here. Is sugarcane yield most affected by the variety of sugarcane or the place it's planted? And one thing we're going to think about is so Montaña Verde and Los Tomarandos are the two places where you have the highest average yield. Let's go back up and look at those places. So those places are here, this one, and this. Wait, I forget. Oh, I'm so tired. Montaña Verde and Los Tamarindos. Montana Verde, and Los Tomarindos. So these two places are the places with the highest yield of sugarcane. Um, you might have some predictions about why that might have been true. Um, and it turns out that there really are just some places where there are more nutrients in the soil, or there might be more or less extreme drought or extreme flooding. But you can see there are two places where it doesn't matter. You can plant all the different types of sugarcane, and some of them are going to show up. Similarly, that first type of sugarcane, the genotype that's V99245, that mutation makes it so there's less sugarcane. So your answer down here secretly is it's both, right? It's both. There is a difference for yield for each location, and there's a difference for each genotype. Now, the last thing you're going to answer is recommending varieties to farmers in each of the 5 regions. So now what we're going to do is make a graph where we organize things by the variety and look and see which variety grows the best in different places. And so to do that, I think what I'm probably going to do is when you come into this document, there's going to be a third, I can't see anything. There is going to be a third tab, and it's just going to say final graph data. Thanks for bearing with me as I make this thing. And I'm just going to copy and paste that same graph data again. It's not going to have any of the supports it had before. You're going to see now you've got genotypes and locations, and you've got those different places. Now, what we're going to do is make one big bar graph. So you're going to be able to see not just averages, but like all those different data together. So I'm making one big bar graph. And hopefully you can see now that it's by gene. And then the different colors are going to tell you about the different places. So I'm going to find it helpful to hit Control C in this assignment. And even though, moving my stuff around that I can see, just going to go ahead and put this graph directly in. You don't have to do that, but it is going to help make sure, whoa, what's happening? I can't see. I can't see. I bet it pasted. Let's find out. Hang on. It didn't paste. That looks fine. Great. So this graph is going to help you figure out for each region which genotype has the highest level. So we're going to start by looking for the highest blue bar because that's for Santa Lucia. Let's say you're advising the farmers in that area. Which of these genotypes would you plant? Does it matter? Is there one you're definitely not going to plant? Right. All of these blue bars are like similar. There's one that's definitely the highest. There are two, they're definitely higher than the others. I think V918 is probably going to give you the biggest yield, and you shouldn't plant V99285, right? So you're going to say what variety you're going to plant, and then you're going to describe the thinking I just went through as you make the predictions for each of those varieties. One more time in this graph, each of the regions is a different color. And so, like, for example, let's skip to Los Tamarindos. One of those is way, way better for that area than any of the other ones. And so you'll make that prediction. But here you're just going to describe generally how you make that prediction. That's the assignment. When you're finished, make sure that you've turned it in on Canvas. So that's what we're doing today and tomorrow. I hope you got a little bit of practice with me. You know now how to use this to make an average. And I look forward to seeing your graphs. You are going to have to go through the steps I just made to make your own graph. So even if you're working with somebody, you do need to have your own spreadsheet. All right, that's it for me. I hope you're doing well out there. See you soon.